Hi, and welcome to the Cerebellum series. And today, I'm so excited to tell you about Lubica Kubikova, who was born in Slovakia. She received her PhD at Comenius University, where she studied dopaminergic control of behavior in poultry. Then, during her postdoctoral studies, she came to the United States and she studied with Erich Jarvis at Duke University, focusing on the dopaminergic system in songbirds and function of the striatal vocal region for the learned songs. One of her key discoveries was that in the male zebra finch, singing to a potential mate coincided with dopamine release, which was significantly less when a similar song was sang when the bird was alone. After her return to Slovakia, she joined the Slovak Academy of Sciences and established her own research group focusing on neurogenesis in the brain and particularly regeneration of neurons after injury. She also serves as the head of the Department of Physiology and Ethology at the Centre of Biosciences. Her recent work, the one that has really interested me, shows the role of the cerebellum in learning temporal and acoustic features of the bird song, something that is rather unique, I think, in that community. Lubica, thank you so much and great to have you.
Yeah, thank you very much, Reza, for the introduction and for the opportunity to talk at this forum about our results. I work in rather small group of scientists, and we focus more on neurogenesis and neural recovery, as you said, recovery after brain damage. But somewhat accidentally, somewhat surprisingly, we got also interesting results on cerebellum, and I'd like to share that with you guys today. But first, I'd like to give you some small introduction how we got to these results. The human speech and the bird song share many similarities. One of them is there are some similarities on the brain anatomy, and here in white you can see the brain pathway that is necessary for the speech learning. It connects cortex, striatum, thalamus, and cortex back, and similar pathway we can find in the songbird brain. The cortical region here, the striatal that is called ARIX. I will be talking about this a lot today, and the thalamic region back to cortex. Similarly, in humans and songbirds, we have this motor production pathway that connects the laryngeal motor cortex via several brain regions to brainstem nucleus ambiguus, and these motor neurons then innervate the larynx muscles. Similarly, in songbirds, the signal goes from HVC to this nucleus 12, and the neurons innervate the respiratory and the vocal organ syrinx muscles. So first, we work with these birds that are called zebra finches. I'm sure you saw them many times already, and we were interested in the function of this nucleus area X within a striatum. What's the function of this nucleus in adult birds? So we lesioned this. This was the work done by my former PhD student, Eva Bosikova. She lesioned this nucleus using neurotoxin. And here is the image, immunohistochemical staining of the neuronal marker, a coronal section with the two nuclei. 1 day after the toxin, after the lesion, you can see the lesioned area, but surprisingly, 1 month, 3 months, 6 months, several months after the lesion, the area recovered significantly, as you can see here on the graph. At 2 months post-lesion, the lesion is already less than 20% of the original size. And we checked this also by the tunnel staining. Tunnel staining shows you the apoptic cells in the area, and you can see that the neurons there are actually really dead. And then we asked, so how is this recovery done? Is it that the neurons from surrounding area invade this lesioned area, or is it due to neurogenesis? Because adult songbirds have neurogenesis, and the new neurons come also to the striatal area. And we performed this double coating staining. Double coating is a marker that it's a protein that is expressed in neurons after the birth for up to 30 days. So it's a marker of newborn young neurons. And we found that in the lesion border, there is a high number of this double coating labeled new neurons, and the area is recovered by neurogenesis. We also injected this BRDU, it's bromdeoxyuridine, it's external marker, it's thymidine analog, and it stains the newborn cells, and it was colocalized with this other marker that is known to be expressed in medium spiny neurons. So we know that these neurons that are recovered are medium spiny neurons, which is the most abundant cell type within area X. And it's also known that area X expresses, the neurons within this area express this gene EGR1, its transcription factor. And the gene here is expressed only when the bird is singing. It's singing-induced gene. When the area is lesioned, of course, there are no neurons, there is no EGR1 expression, but at 1 month post-surgery, already there is some significant recovery, and already we see some EGR1 expressed in some small area here. At 6 months, it's even bigger area that expresses this gene. And as you can see in this graph, the more the bird sings, the more of the EGR1 is expressed there. So we found that it's functionally, so the new neurons that come to this area are functionally connected to the circuitry that controls the vocal production. Not only, or these changes in the brain were associated with some behavioral changes. This is how the song of the zebra finch looks like. It sings a sequence of syllables. And when there is a repetition at the end of the song, small repetition of the last syllable, then the bird, after the surgery, repeats the syllable even more often. This is undirected song, when the bird is alone and he sings alone. And here you can see it on the graph that it occurs at around 1 month, and the repetition increases in course of the experiment for up to 6 months at least. It's the red line in comparison with the blue controls. And like this. And when the bird is singing directed song, it's these two sonograms. The song is accompanied with it's a courtship song. It's accompanied with the dance, and then the bird is singing even higher number of the repetitions, and it looks like it sounds like he's stuttering. I will play you example of this song behavior. This is before surgery. So So he was repeating at the end, and this is 3 months after surgery. So you could see the stuttering-like behavior, stuttering-like song. So the bird seems that he has a problem to connect or to continue with another motive of the song, to continue with the song. So then we wanted to know what other regions are affected with the lesion of area X. And this was done by Kristina Lukacheva, that was also my PhD student. At that time, she went to Belgium and worked with Julie Hamad, that was PhD student there, and they teamed up and performed magnetic resonance imaging of these lesioned birds. So here in these pictures, so each of these images is average scan for 12 birds. And you can see the lesion in area X it's hyper-intensive signal here due to edema in that region. And here with black, it's the affected area, the area affected by the edema. Then they performed what's called diffusion tensor imaging. And this is a method, it's a special method of magnetic resonance imaging that is performed, the scanning is performed at several time points, in this case for up to four months. And they found what areas and when were affected. So we hypothesized that area X will probably affect this thalamic region DLM. It's the dorsal thalamus. And yes, they found the effect on the thalamic region. They also found effect in the region surrounding the lesion. They also found effect, which is not seen here, but it's in the nucleus HVC, it's this structure. But what was the most surprising for us, that they found significant effect in the cerebellum. Cerebellum was not, and Teresa mentioned that it's not usually seen as to be connected with the vocal circuitry. Therefore, it was very surprising for us. And the affected region was the lateral cerebellar nuclei. We then asked, what are these changes? Because we found only when was it that, when were the changes and where were the changes, but what are the changes? So we first just calculated the number of cell nuclei in these regions, HVC, the thalamic region, and the lateral and medial cerebellar nuclei. And we found that even in the kind of faraway regions, as the cerebellum, the cell counts were decreased significantly, even for several months after the surgery, after the lesion. Since we found the changes also in HVC, we hypothesized that this decrease can be due to decreased neurogenesis, because HVC is known to receive new neurons in adulthood. So we injected again the BRDU marker and found actually the opposite, that in HVC there was actually at 8 days post-op and at 2 months post-op, there was increased number of new cells. These include glia too, but these are in colocalization with the neuronal marker. So even in this area, there is at 8 days and 2 months increased number of new neurons in this area. So at least this decrease here is perhaps later compensated by the increased neurogenesis in the area. We also assessed or examined expression of other genes. One of them that was interesting for us was FOXP2. It's a fork head protein, and it's associated with it's highly expressed during song learning, and it enables, or the higher levels of FOXP2 enable neuronal differentiation, and also they're associated with the spine density. And we found that at 8 days, so 1 week post lesion, there is increased expression of FOXP2 in HVC in the thalamic region and also in the cerebellar lateral and medial nuclei. And we found biphasic effect or increase also in cerebellum and in the medial cerebellar nuclei. We think that this reflects some massive synaptic remodeling of these regions at these times. Then we also examined expression of PNNs. PNNs are perineuronal nets, and they are highly expressed when the song is learned at the end of the song learning, and it stabilizes the extracellular matrix. And we found here that there is higher expression of perineuronal nets, especially in HVC, not so much in cerebellum, only at one time point, but mostly in HVC. We also found later on that perineuronal nets have also some protective effect against harmful agents. So this is probably the role of the increased perineuronal nets in HVC at these for up to 2 months post injury. When we published these results, also other groups came with their results, because it was interesting that cerebellum is somehow involved in the vocal learning or vocal production. So Nicholson at that time published a tracing study, and they injected the dextran-amine tracers to the lateral cerebellar nuclei and found so that the dextran-amines were conjugated with different colors. Each row is different bird here. So they injected to, they injected to the lateral cerebellar nuclei and found the tracer, the axons in the thalamic region DLM and in the surrounding thalamic zone, contralaterally. So up to now, it was known that area X from here projects to the thalamus, to the DLM nucleus here, but they found that also DLM and the surrounding thalamus projects back to area X. And this is how cerebellum is connected with the vocal circuitry. Here so here they injected dextran-amine, here they injected lentivirus, and they overexpressed GFP or M-Sherry. They injected to the thalamus, and they found the tracer or the GFP in area X. They found the synaptic field in area X. Similarly, in the same year, Pidu and co-authors also injected tracers to area X, to the cerebellar, lateral cerebellar nuclei, or the cholera toxin to area X, and found overlap in the dorsal thalamic zone, overlap of the cell bodies that project to area X, and the synapses that are in close proximity to the cell bodies within this area. These authors also electrically stimulated DCN and measured the activity of the neurons in area X. So here are different activity of different neurons in area X. And you can see that after the electrical stimulation of the cerebellar nuclei, it induces peak in activity of different neurons with the latency of the response for up to 50 milliseconds. It is known in humans that during speech there is activation within cerebellum in the cerebellar nuclei, and the malfunction leads to speech deficits, and these deficits involve timing perception, articulation, phonation, loudness, and also tempo, and the people can either accelerate or decelerate on the speech. And lesion can induce also stuttering or cause stuttering and change temporal parameters of the speech. However, it was not known what is the function of the cerebellum for the learned birdsong. So PIDU again, this group performed this study in the juvenile zebra finches, and they lesioned DCN. This is horizontal picture of the brain. So they left the medial cerebellar nuclei intact and lesioned the lateral cerebellar nuclei using electrical current. And they looked at song learning. The song learning, the zebra finches learned the song during the sensory period. It's from about day 15 post-hatch, and they only memorized the song at that time. But at around 45 to 50 days, they start producing and imitating the song. And that's what a little bit after that, they lesioned the cerebellar nuclei during the period of song learning. And they compared the similarity of each syllable that was learned from the tutor. And they found that the similarity was better, or the imitation was better in the sham lesioned birds or sham operated birds than in the lesioned birds. And when they looked at the similarity score in comparison to the lesioned area, this is intact DCN area. So this bird has the biggest lesion. So they didn't find really some linear relationship. The bigger lesion, the bigger effect of song. It was it didn't depend on the size of the lesion, but the similarity of the syllables was impaired. Since, as I told you, cerebellum also affects timing, so they looked also at syllable duration. So they measured duration of each syllables in sham-operated birds, and in the sham-operated birds, with black here is shown the distribution of syllable durations of the birds. 5 or 6 days after the sham operation, the distribution of the syllable durations is shown by red. It's not very different, but as adults, these birds have they shorten the syllables. However, the lesioned birds, this is the distribution of the syllable durations originally, and when they have the DCN lesion, I'm sorry, here, the change here in the syllable duration is bigger than the change in the sham operated birds. Okay, and when they looked at adults, the sham operated birds changed the syllable duration more than did the lesioned birds. The lesioned bird didn't change the syllable duration, or only a little. Since it's known that also pitch of the sound of the syllables is a learned feature, they also measured pitch in these birds, but they didn't find actually significant differences in pitch. We performed similar study, and this was work done by my other PhD student, Rebecca Radic. She lesioned the birds, but this time using the neurotoxin. She lesioned the DCN. She first recorded the songs pre-op, and then for up to 4 months post-op. And so she performed the lesions. At 5 days, she performed magnetic resonance imaging just to prove that the lesion efficiency was okay. And then at 4 months, when she sacrificed the birds, she also proved that using immunohistochemical staining. So she lesioned the lateral cerebellar nuclei, not the medial one. And then she analyzed the song. And she found that the birds sang longer songs. Almost immediately, they started singing longer songs almost immediately after the lesioning. And the songs were longer because of this introductory part that contains only introductory notes, but also this part that follows, and it contains the learned syllables, and the syllables are organized into the sequence of song motifs. So both of these parts were affected, and this introductory part contained more syllables because the prolongation, it could be due to increased number of syllables, but also the silent intervals, the silent gaps could be wider. But she found that the birds sang more of the introductory notes, and they sang also more of the syllables afterwards. And the syllables were organized into motifs. So she found also more motifs within the song. Then she looked at tempo, because tempo is also affected by cerebellar malfunctioning in humans, and she found that the song tempo, song tempo she assessed by measuring the motif. It's the sequence of syllables that is done, that is given. So she found reduced duration of the song motif, suggesting the tempo is increased. And now, why is the tempo increased? Because they sing faster the individual syllables, or they shorten the gaps between the syllables, or there is some combination of that. So she found that they sing the syllables faster actually, but they don't change the silent gaps. The silent gaps are actually from about 20 to 50 to 60 millisecond long, and the syllables are from 50 to 300 is maximum, probably, milliseconds. And at the end, we looked also at some acoustic parameters of the song, and we found that immediately after cerebellar lesions, the birds decrease amplitude of their singing for the following 3 days. At 2 weeks, it came back to the baseline levels. We looked at the pitch. We also, as with the young birds, we also didn't find any changes in the pitch, but we found there are some syllables that are harmonic. They have this harmonic structure here, and pitch is actually the fundamental frequency of the first harmonic. And when we measured this part of the syllables, we found a significantly decreased the fundamental frequency of these syllables. So we found a transient decrease in amplitude and long-term decrease in the fundamental frequency of some syllables. So they sank deeper, more deeper, these syllables. And to conclude these results, it was found that cerebellum in juvenile songbirds is necessary for vocal imitation, as was shown by the syllable similarity. And it's necessary also for syllable duration during the learning. And in adult songbirds, it affects temporal features of the song, such as song duration, especially the number of elements and tempo of the singing, but tempo of singing of the syllables, but not the silent intervals. And it affects also acoustic features of the song, such as amplitude and fundamental frequency. I think that's it, what I wanted to show you. And thank you very much for your attention. And I'd like to thank to Kristina Lukačová and Rebecca Radic that performed these cerebellar studies. Laco Bačiak helped with the magnetic resonance imaging in Slovakia, and also Julie Hamait at University of Antwerp that performed with Kristina the diffusion tensor imaging. Actually, these were the first results of cerebellum that we found. Thank you very much. If you have any questions, I will be happy to answer.
Thank you, Lubica. That was wonderful. I learned so many things I didn't know about. And if anybody has questions, please just go ahead and raise your hand. I'll be happy to call on you. But I'll start. So one of the really surprising things for me was that you demonstrated that striatal lesions recovers substantially at 6 months. And I'm wondering that neurogenesis that you showed. Now, what about the cerebellum when you damaged it, the DCN lesion that you caused? What do you think? Is there anything happening there as well?
Yeah, we checked. It doesn't recover. It doesn't. And you can see that I showed you some pictures. We checked the lesion immediately after 5 days after performing the lesion with MRI, but at 4 months we performed immunohistochemical staining and the neurons were not there. So there is certainly many of BRDU cells, which are, however, glia. It's not the neurons.
Yeah.
Yeah, we wanted to know whether cerebellum also can recover, but it seems not.
What's
And
the difference? What do you think is the difference between striatal system and the
well,
cerebellar?
it's known, the new neurons come from neurogenic zones. And there are two zones in mammals. One zone is around the third ventricle, and the new neurons in rodents, they go to mostly to olfactory bulb. In humans, it's controversial. People argue still. Some show that neurogenesis in humans occurs in adulthood. Some show it doesn't. But if it does occur, they showed new neurons going actually to the striatum, which makes sense. It doesn't go to the olfactory bulb because, of course, for rodents, it's the dominant sense, but not for humans. So in this aspect, the songbirds are more similar to humans than the rodents are. Another zone where the new neurons come from is inside the hippocampus. So both of these regions, the neurogenic regions, are in telencephalon, and they don't go lower than to telencephalon. They migrate only within telencephalon. There are some studies that show that the new neurons migrate to hypothalamus, but not further down. Unless there would be some, I guess it's quite far away.
One of the interesting things, surprising things, I thought, for the DCN lesion was that you found that the syllables are faster. The zebra finches sing the syllables faster, but not the silent parts,
Yeah.
the silent gaps. But at the same time, there was reduced amplitude of the acoustics. It was surprising that at least to me, that the song became faster syllables.
Yes, and it's actually shorter time interval than people can distinguish. Even that was the reason why I didn't play the songs, because we cannot catch it with our ears. The very small differences. In humans, it was shown that people with cerebellar impairment have difficulties to distinguish short time intervals. But the short time intervals means several hundreds of milliseconds. So I think I even wrote it down. 10 to 15 milliseconds, they were unable to distinguish. But usually the studies are performed using several hundred milliseconds, some kind of, in that kind of time scale. And I think the birds are just much more sensitive, and they can distinguish much smaller time windows than people can.
So do you see this as a learning effect, that the cerebellum is, so that the damage to the cerebellum then induces a change in how the animal can learn the song, or do you see it in how they can produce the song?
No, I don't think this, what we found was not the learning effect, because they don't learn in adulthood.
I see.
What the other people showed, it was learning effect. But I don't think what we showed was the learning effect. It was probably the effect of the missing signal from cerebellum.
I see. Janner, you have your hand up. Please go ahead.
Yeah. Hi, thank you.
Hello.
It was a nice talk. Thank you so much. I was interested in the time course of the effects that you were mentioning. I know that you were showing some plots, but maybe I was missing. So after these cerebellum lesions, there was changes, in the tempo, in the pitch, in the fundamental frequency. And I was wondering, which is the time course? All these changes appear around the same time point, or is like a
No,
consecutive
like
change?
here, the amplitude is reduced only immediately after the lesioning. So we measured it first, second, and third day, and then at 2 weeks. So at 2 weeks, it already came back to normal.
Yeah,
But
that's
other changes, like this fundamental frequency, it occurred immediately and stayed reduced for the whole time.
yeah, I see. Thank you so
Okay.
much. Yeah, and I was also wondering if you have seen, because the cerebellum has been also, at least in humans, related to the timing processing on the scale of seconds. And so I was wondering if you have been found any changes in the sequencing of the sequences that the animals learned, like A, B, C, D, and then they start making random
Yeah, we
mixes.
checked that, but they didn't change the sequence. They don't change the sequence. Also, I think it requires maybe different model to look at changes in sequencing, because zebra finches are very fixed. After they learn the song, the song in adulthood is very fixed, very crystallized. So even these changes, it's kind of surprising that we found these changes, but I think there will be better models to look at changes in sequencing. And it would require a bird, as Bengali finch probably, or some other bird that has more variable sequence naturally in adulthood. Yes. Because once it's so fixed, it's very difficult to change the sequence in zebra finch. But we did look at it. It didn't change.
Okay, thank you so much.
Yeah. But it's also true that they sank a lot of these pieces, like, as I said, they have fixed sequence, which is motifs. It's like this. It's sequence of these 5 syllables in this bird, but also they have sequence that is not, it's not the full motif, and they have also quite large number of these syllables that don't form the motifs. So that's the only thing that.
Okay. Yeah, thank you.
Thank you, everyone. Are there any other questions? Lubica, thank you so much. I thought that was just wonderful. I learned so much from you today.
Thank you very much too for inviting me and for the opportunity. Thank you.
Bye-bye.
Bye-bye.